Hello again, children. We have got to stop meeting like this. No, but really, we've got to stop meeting like this. I am so sorry. I keep getting pulled out to go to meetings and practice tests and teacher stuff and whatnot. And believe you me, it is stressing me out missing all this time with you guys. And I'd give anything just to be like, leave me alone. But seriously, I do miss you guys. Everyone except Lee, Green, and Hutcherson. I don't miss you three, but everybody else I do miss. You three, not so much. Okay, let's get started. My fist is so crooked, which is relevant to this chapter, actually, because we were talking about protein synthesis, mutations, and everything else. So, everybody get your little highlighters ready. Oh, hello. And let's begin. All right, first things first, I'm the realist. Second thing second, sorry about that. So I want you to understand the difference in these things, okay? The difference. And the order of them, okay? So first we have the cell, and the cell is the nucleus. In the nucleus is the chromosome, and the chromosome is the DNA, and within the DNA is the gene, okay? Very important. One other thing, I want you to know how the DNA makes the gene, and it is the way that the nucleotides are arranged. I'm just writing on top of everything, whatever. So remember, somewhere in your notes you should have it. I don't have it in my notes because I just kind of wing it in class, but if you recall, in class we were talking about AGT could mean black hair for me, but ACT could mean brown hair for you. So the way that these nucleotides are arranged, whatever order they're in, and then, CAT could be blonde. So that determines what genes you actually. If you need to pause this video, you're more than welcome to it. Alrighty. You kind of already know how these get passed down to offspring, so let's move on to transcription and translation. Now remember, memorizing this will help you tremendously, because if I ask something like, oh, what's made in translation? You know that it's going to be over here, protein. Memorize this. I promise it will help you, okay? All right, transcription and translation. Transcription, where we read the DNA, it's obviously going to take place in the nucleus, right? Because that's where the DNA is. By mRNA, super important. Translation, where we build proteins. Who builds proteins in the cell? The ribosome. So definitely know that. Just remember, how do we make a script? We write it or copy it down. Yes. Our biggest thing, tag C and UAG C. And of course, you need to know the differences in DNA versus RNA. DNA is like this, RNA is like this. DNA has a T, RNA does not have a T, it has a U. Okay, this is my biggest thing I'm going to want you to pay attention to. Read. Alright? Because if I am asking you what's going to come out of what, I could be talking about two different things. If I'm referring to transcription, I'm going to be talking about this little number. If I'm referring to DNA, I'm talking these two pink things. RNA is this blue thing. I want you to know, here's something I'm going to ask you, okay? I'm going to be like, in transcription, what would AGT turn into? Well, transcription is where we get our mRNA, so this would be the answer for that. But then, what if I ask another question that says, in translation, what would AGT turn into? Well, translation is turning all of this into a protein. So the answer is going to be different. The answer is going to be whatever UCA is according to our codon chart. Definitely know how to read the codon chart. First, second, third. It's fairly simple. On Monday, I will have this on the board for you to be able to read. So don't freak out about memorizing this. I would never be that cruel. I have been known to be evil, but not that evil, okay? Make sure that you understand how to do this, okay? If DNA says this, what will RNA be? What will the protein, the amino acid be? Okay, I'm gonna need you to know how to do this from start to finish. All right, mutations are mucho importante, okay? There are point mutations, which is the substitution, insertion, and deletion. But keep in mind that only insertion and deletion are known as frame shift mutations. Reason being is because they change the whole thing, whereas substitution only changes one little thing. I want you to understand why substitutions are usually harmless. Usually, not always, but usually. And I want you to understand why frame shift mutations are a lot more dangerous than substitute mutations. And here we have all of our throwback questions that are going to be on there, okay? Anaphase versus anaphase 1 versus anaphase 2. These are three completely different things, okay? This one is mitosis. Both of these are meiosis, okay? I want you to know what the difference is in all these. Rudolf Virchow, know what he discovered in the cell theory from way back in the gap. Prokaryotes and eukaryotes. Prokaryotes are, bacteria, and you are a eukaryote, and so is Gertrude and Frank and that cute little dog that's always running around campus. What organelles do they share? Okay, obviously, things like even though we don't have a cell wall, Gertrude is a eukaryote and she has a cell wall, so that's something that both her and bacteria have. But then there's one super important thing that prokaryotes do not have, and that's what I want you to focus on for that question. Hypertonic, hyper, this too much, this a lot of salt. This is red bottoms, this is muddy shoes. Anyway, hypo, as big as a hippo. And iso is isite, okay? I'm going to need you to know what happens to cells in each of these. Viruses in the lysogenic cycle versus the lytic cycle. How do they infect cells using both of these methods? I want you to know how enzymes react according to pH, temperature, and the concentration. I want you to know how to do a dihybrid cross. That is the big Punnett square. And I'm going to come back to that in just a minute. I want you to know mitosis diploid versus meiosis haploid. What does that mean? It means how many cells does mito have? 46, right? How many cells, did I say cells? I meant chromosomes, dagnabbit. Okay, anyway, how many chromosomes do if I only have 46 cells in my toe, that's a very small toe. But that's aside from the point, that was my bad. Okay, so how many chromosomes do myovaries have? Well, myovaries, the eggs in myovaries, only have 23. Okay, so that is what a diploid versus a haploid is. Remember, a haploid is only half. The phospholipid, what's it look like? And finally, active transport. In case you couldn't tell, I changed my mind several times about what the last question would be. but I figured it's important to go back, way back in the gap to active transport and know the difference between channel and carrier protein. Okay, I feel like this must be done because I saw a lot of people make this mistake and it just got a little crazy. So I figured I'd do a quick demo. Oh, that's cool. This Punnett score is way off, but whatever. Okay. So let's say that mom is heterozygous for brown hair. Okay, she's got brown and blonde in there. Brown and blonde. And she is homozygous recessive for freckles. This means she has brown hair and does not have freckles. Now, combine her with dad, who is blonde haired, but he is heterozygous for freckles, so he does have freckles. Okay. This was something a lot of people got messed up is in the setup. Okay, so remember, at first, and only at first, B goes with the F. Okay, so big B, little f. And then B goes with the other F. Okay, so big B, little f. BFF. And then, after we're done with him, we take the little b and do the same thing with him and both F's. Okay, so mom is done. Dad, we do the same thing. Little b, big F. All right, we're just going to keep it in order, okay? B comes first, F comes second. So let's just keep it that way. Little b, little f. We're done with him. Let's do the second one. Little b, big F. Little b, little f. All right. Now, this is where a lot of people got a little crazy, but I just want you to know that, remember, mom and dad, both of them had a B genotype and then an F genotype. We have to put it back in that order. So you bring down the B and put it with the B. Okay, and then you do the F. A lot of you mess this up whenever you're setting up your dihybrids. Remember, just like mom and dad, mom had the B's and then the F's, dad had the B's and then the F's. So B's and then F's, okay? And you do that over, okay? And you keep doing it and keep going on like that until you get done with this whole thing. All right, remember, it's very important that you set it up just like this, okay? Just like the way that you had it here, B first, F second, you do that all the way down, okay? Because if you don't, it'll just throw off your whole thing that you're doing. All right, kiddos, that's it for today. If you should have any more questions, you know you're welcome to message me on Remind. And at this time, go and do what you're supposed to do and let me know if you have any questions. Okay, I love you. Bye-bye.